Sure.
Thank you, Liz. Hi, everyone. My name is Sudha Jairam, and I'm the director for our Center for Global Surgery, and I and a number of our colleagues have tried to create this forum for a monthly global health-related meeting to kind of share all of our work and have it be faculty from across the university. With that, I would love to invite Dr. Mohanty to give us a talk about what he's been working on. I've heard tremendous things about Dr. Mohanty and his cutting edge work on diagnostics that are like based on exhaled molecules, and it just seems absolutely fascinating, and that's coming from a surgeon. So thank you, Dr. Mohanty, for making time to speak with us today, and I'll turn it over to you.
Great. Well, thanks for the invitation. I'm excited to be able to interact with this part of the community of the campus. There's always a barrier. Maybe it's just because we're across the street on how to engage our colleagues up in the School of Medicine better. So what I hope as I go through this is that this will stimulate some conversation and some ideas about further collaboration with not just me, but other my colleagues. So right here, I run this advanced materials and micro devices lab. We call it AM2 lab. And this is the portfolio of stuff that I have in my lab. I'm an engineer, so we tend to try to keep be prepared and be nimble on the funding wings. So the things that relate to global health, of course, are the diagnostics and environmental stuff. I will be talking mostly about the diagnostic aspects. Now, the title of my talk is Stakeholder-Driven Research to Advance Technologies for Affordable Diagnostics in Resource-Limited Settings. So what do I mean by this? Well, ideally, as all of you know, there's a lot of great technology out there, but it doesn't ever reach the end user. And a lot of times it's because engineers just develop stuff in their ivory tower and then go hope that someone will use it. Sometimes there's some business cases, sometimes there's other technology out there. But I'm going to talk about some things I'm working on. So TB is one I'm working on. I do this in Uganda. And I think this crowd knows that TB is disproportionately affects countries of lower socioeconomic status. So certainly United States, we don't see this much, maybe 10,000 cases a year. But if you go to India, African continent, you see a lot. Pneumonia is also a significant one in the United. It affects 2 million annually, and it's the #1 killer of children under 5 worldwide. So I've been talking to a lot of my colleagues about that problem. Now, the first two I have funded, schistosomiasis is another one that I am in my stakeholder stage for diagnostics. And this one, the statistics on this are quite impressive in a bad way. 236 million people are infected annually and this is considered a neglected tropical disease. So this is quite significant. And the diagnostics for these are quite primitive, typically around smear. There are some molecular diagnostics, but they are expensive. And then, of course, malaria, which I'm sure a lot of you are familiar with, 249 million infected annually. And I have colleagues in Benin who have been asking me to help them with this. So this is also in the stakeholder stage. So I'm looking to fund this. I have some ideas, proposals pending on them. But really, I go and I take my technology and I try to solve these problems in low resource settings, at least in this context of global health. So I have a few different technologies that I work with. So up here on the left, I have this nanotube-based breath sensor approach, and on the right, I do a liquid-based, and then below, I have this molecular approach. Using nucleic acids, but using a peptide nucleic acid as the sensing element, and that has some advantages, which I'll describe. So over the course of this presentation, I'm going to talk to you about each of these. Now, before as I get into it, so this particular aspect of this technology is you take an exhaled volatile organic compound. It could also be from urine. could be from skin, condensate. And these are known to be associated with certain diseases. And I'll get a little bit into about why that is. And then we have in the middle here this nanomaterial, TiO2 nanomaterial sensing system. So it's a metal oxide that we synthesize using some simple anodization techniques, and then we functionalize with metals. The whole idea is that you would take something very non-invasive, very fast, and use something like the smartphone to get some diagnostic information, and then ideally provide some information to the physician. Now, before we get to details on that, I want to just sidebar a minute with a little bit about how global health is really hard to do. So all of you, those of you that are active in the global health field, know that it's really hard to do this research. It's very hard to get funded. So I have some slides here. This was from a presentation Monty Pi gave, who's at McGill, and he took it from Gene Weather, Walther. And it's a little bit old, but it makes a point. So a few years ago, a lot of you remember we had some Ebola outbreaks occurring, and it really made the news, right? And the outbreaks that occurred at the time between, say, 1976 and 2015 amounted to about 30,000 cases and 13 deaths. And this was all over the news. There was a great response. Stakeholders came to respond. All of these companies came and they said, we're going to come up with a way to handle this, diagnose this, treat it, right? All this came together, and there was a lot of stuff in the White House. Philanthropists came all over. They had panels and all these tools, right? So this was an impressive response in a reasonably short amount of time when the most recent major outbreak showed up. And then these are kind of diagnostic companies that showed up. And this slide is I took from Monty Pi, who took it from Jim Gallardo at the Bill and Melinda Gates Foundation. So all this happened, and I'm going to talk about TB. So you have a disease that over 40 years killed 13,000 people. In the same amount of time, it killed 40 million people in TB. Yet TB is not funded in the same way, right? The emphasis and the drive to get rid of TB, that kind of gravitas is not there as it was for Ebola in a short amount of time. So I only bring this up as a global health community, whatever problem we're working on, we have to find a better way to sell it, because we're always trying to fund our ideas. And a lot of times we go to these funding agencies and maybe you get an R01 or R21 and you go and you work on it. But this is a very, very slow and crippling way to actually achieve our goals. And I think developing large-scale centers to go after not just TB and other diseases with some intentionality, and actually going after other funding sources than just the federal government, and even and maybe even trying to stand up some foundations that focus on this and cultivate that relationship will help. So that's just to plant the seed of the hurdles, which you all know that we have when we do any kind of global health research and diagnostics. Now let's go to the pain of TB. So if you were to go around the world and take about 100 people in the world, about 1 in 100 have HIV, 1 in 100 have cancer, 5, maybe 6 out of 100 have diabetes. It's one of the fastest growing chronic diseases worldwide. And then roughly about 33 people out of 100 have tuberculosis, latent or active, right? So that is a third of the world has TB. So this is obviously a huge problem. And when you start putting it in these statistics, it becomes quite serious, especially for a problem that has a cure, but we're not getting there. So why is that? So there is some literature published, and this was in 2014, but some of it still stands, is there's a lot of barriers to screening and diagnosis. And here's a list of them. Some is lack of knowledge and not proper training, access. So there's a lot of pieces that are there. I won't go through all of them. I think most of you are probably familiar. But the piece that we're trying to address is the ones ones in circle, the improve diagnostics and try to get better training, and then try to have something cheap so it can reach the patient and get rid of that disparity. One point I want to make about this slide is that this problem of any disease in general, but TB, will not be solved by simple engineering solutions, right? This is a total, needs a total effort by all the stakeholders, physicians, public health officials, everybody's there. So a lot of times technology will come out of a lab and they think they'll just solve the problem. And we see time and time after again, this doesn't happen. We still haven't solved the malaria issue, and we threw a ton of money at it. We still have malaria, right? So there hasn't been historically, I think we're getting better at it now, a good partnership between engineers, physicians, public health managers, everybody involved to do this. So if we go look at TB specifically, you know, these are the standard ways: X-ray, acid fast smearing. We've been doing that for 100 years. You can see that the sensitivity is all over the place. Gene expert, really great, but need infrastructure. And it's still sputum based, and then sputum culture, really great. And you can do it, the culture in, you know, a week to 8 weeks, depending on if you have a fast midget or you have to do regular approaches. So these are the approaches. One is just simply a visual X-ray. The other ones are all sputum based. So the WHO says, well, if we're going to solve this problem for TB patients around the world, well, we really need a triage test that's low cost, and we need something non-sputum. So one stat, which I don't have on here, is that when 5 people present for TB, only 1 of the 5 actually have TB. The other 4 have something else. So this is a large burden on the healthcare system. So we went through this, we started understanding this, and by using the WHO TB target product profile, we use that as boundary conditions to help develop some tech to address this problem, which then can be broadly applicable to other aspects. So let me talk about my stakeholder engagement. So when I was in grad school or even undergrad, they never taught us about stakeholder engagement. It's only now we're starting to do that, right? So it's super important, even before you start to make your product or your research, go talk to the people that experience the problem every single day. So these are the stakeholders I engage. So the physician, obviously, the individuals who treat the disease in different clinical environments. Some in the hospital, some rural, you talk to them all. You talk to patients who've experienced this, and they'll tell you their experience in being diagnosed, how far they had to travel. You talked about lab technicians. Lab techs, when I found out in Uganda, they're super overworked. Probably true here too. They get so many samples, they have to operate it unless you have a ARUP scale automated lab to take samples, which we do here, but they don't in a lot of places. The lab techs are overworked and can be prone to mistakes. And so you want to think about how your diagnostic influences them and how you can work in their healthcare. Then, of course, we have the public health managers for a given community. A lot of times it's a TB manager for an entire country. And they actually make the choice of which test to use based off the performance data. And of course, the nurses, a lot of time, the nurses are the ones that are actually interacting and getting the samples. So you need to understand how the technology would influence them and what they would want, right? And then hospital CEOs, how do they choose? Regulatories. There's all these aspects of this, including a diagnostic manufacturing of a test. If it's not easy to manufacture, performs well, that's still a problem, right? So this is a big complex problem that we go through. So we've actually gone through all this for TB and for other diseases too. And PIs don't often do this, but a PI should do this along with support from students and experts in the field to ideally come up with, if you look at this this chart on the right, you've got, ideally involve them extensively, tall order sometimes, but you try to do your best. So you engage them. So there's an element of co-design when you do this. Now, so this is me collecting breath off a rural clinic, off the road, about 15 feet from a crowded road from a TB patient. So these students would come in, or these patients would come in, and we'd all wear these N95 masks, and I would collect breath and I would take it for processing after. So this is the kind of the experience I think everyone should go through. Now, obviously, I'm not going to treat patients, but understanding and experiencing what it is a healthcare professional goes through and seeing what the patient goes through is super useful. So I've been doing this in Kampala, at McCarry University, in collaboration with UCSF for a number of years now. And we've had a lot of good successes, and we learned a lot along the way. Now, we are using this idea of volatile biomarkers for disease. So I have here a picture of Joy. Maybe some of you have seen her or seen a documentary about her. So she is capable of smelling Parkinson's and TB and other diseases. And she has basically hyperactive olfactory cortex. It's a known condition. It's very rare. In her family, it affects the women, not the men. And she had a really interesting story. I had a chance to hang out with her for a week when I went to Tanzania to look at some of the work done with pouch rats for looking at landmines and TB. And she had this interesting story. So she was a nurse, and in the 60s and 70s, she knew she could smell disease, but how are you going to tell someone back then that you can smell the disease with any good diagnostics? So she would have to figure out how to get the doctors to run the test without telling them why. It's like having a superpower that you can't tell anyone about. And maybe super smell would not be my first choice of a superpower, because unfortunately, as you imagine, you smell everything, good and bad. So she knew about this, and then she got married one of the doctors and that she was working with, and 10 years before his diagnosis of TB, she noticed that he smelled differently but didn't know why. And she put it together when she went to a Parkinson's conference for support of other people who have Parkinson's, and she noticed everyone smelled the same. And then that clicked together, and she could smell Parkinson's, which is interesting because, I mean, it's a neurological disease, it's not even an infectious disease, right? So I bring this up to say there's a lot of premise and data to support odor or chemical odors associated with disease, right? And she's a really good example of this. And I can tell you lots of stories about her. I know we're short on time, so I won't spend any more time on it. But when we look at this, we're looking for one of the biomarkers I look at are these volatile organic biomarkers, right? And the data behind this is quite strong. Here's a bunch of diseases that have been documented for odors. We don't use any of them for diagnosis yet. Part of the problem is we don't have point of care sensors to pick up these odors. Not yet, but I mean, I have some research types ones I'm going to show you. So that's part of the reason why. Otherwise, you have to do these on a mass spec, and then you have to decide, is it worth it versus other clinical symptoms. So, but it's a non-invasive methodology when you think about something like a TB. So if I have these breath VOCs, this is what a normal concentration of breath VOCs would have of these various chemicals. Probably worse for us on a bad inversion day with all the chemicals, but it gives you an idea, right? So then in this particular case, the Mycobacterium tuberculosis has these biomarkers that are their metabolic byproducts. So this is their odors that come out as part of their living. Smells sweet. So this is nice because what that means is that since your body doesn't produce these, they're exogenous. And then this was demonstrated. So bees were demonstrated to pick this up, and then the pouch rats at the bottom. And that's me hanging out with the pouch rat. I wasn't in this publication, I want to be clear, I should be. But I got to take a picture with a retired pouch rat, and I got to watch these pouch rats get trained. Still a sputum-based test, but it's fascinating. It really tells you that there is a good scientific basis for this. So conceptually, what are we doing, right? Ideally, I have this point of care breath diagnostic, and it runs over these nanotubular sensors, and I get some data from my smartphone or laptop, and then I can even map it if I wanted to. There's all these things that you could potentially do, right? So ideally, it's antibody free. It is label free, and you're just using inorganic metals and these chemicals, and so it becomes very attractive from that perspective. Now, it's not perfect, right? I'll be first to tell you how my sensors do well, and I'll tell you how I can make them lie to you. Sensors always lie to you. You have to always be skeptical when you read them, even in the clinic. Now, how do we make these? Just for those of you that might be interested in the synthesis method. So I take a piece of titanium, and what I do is I put them in a liquid acidic bath, and I apply potential, and it's called anodization. What it does, it puts an oxide on the top. And then my solution has fluoride. So if you're a material scientist, you would know that fluorides etch oxide, and the ions make these little holes, and they anodize more, but the ions etch more, and it's this tango dance till I eventually get this substrate, right? I made this, it took me 30 minutes. This is when I went in the lab and did stuff. Now my students do it, but it takes 30 minutes to make this. It's really cheap to make. I then functionalize these to give them some specificity. I'll talk about that in a minute, but it gives you an idea of what you can make. Now, you can take this and you can pattern them. You can adjust the size, the length, diameter by just modulating our anodization conditions. So it's it's a nice tunable substrate for a lot of these sensors. And then I can even make a U logo. Yes, I am that guy who made a U logo to get a publication out. So what's interesting about this is that, these are all made of nanotubular structures that are, 100 nanometers in diameter. So it's a very useful technology. So if you think about we're in the clinic when I put this, I could put this on a wire, I could put this on a mesh, and I could put this in a ventilator. So people that might get pneumonia or ventilator-acquired pneumonia, you could throw this in there and look for those VOCs that show up when the bacteria show up. So a lot of ways to pick this up. A little more on the fundamental science of how we figure out what metals to put. So you guys aren't molecular modeling people, so I'm not going to bore you with all that, but I'm just give you basically the 30-second explanation. What we do is we take a metal on the periodic table and we model the molecule we know, in this case, like methylnicotinate, methylpianosate, and we run it through molecular modeling, which will tell me which one of these metals will react with this organic molecule in a thermodynamically favorable way, right? So if you remember your Gen Chem, like you get a negative gives energy, that means it's going to be more likely to occur than others, right? So that's what we do with a few other aspects, and we figured out what metals work well. So I'm not showing everything. We published it. I'm showing you some examples. Then cobalt, chromium, copper, they all work to a varying degree. But so we then we say, well, which one of these can I engineer with? Just because, I remember, I think chromium came out as a very, very good metal, but it's toxic. So I don't want to necessarily engineer with it, right? So I choose some stuff I can engineer and get good results with, okay? So how does it work? Well, I have this nanotubular structure, and what I do is I'll functionalize the metal. In this case, I put zinc, and then I apply a bias voltage on this, right? And then I read current. Then what happens is I have a molecule that comes through, and as it comes through, it binds. When molecules bind, they're really exchanging electrons, right? That's what's happening on the orbital level. Now, this is under a bias condition, so when those electrons, the new electrons show up, well, I can detect those as more current. That's how I know. This is one way of doing it, not the only way. But this tells you how, this tells you if that chemical binds. Now, there's additional specificity because the chemical is known to be, in this case, in this example, that zinc prioritizes the binding of this, right? There's some other things you have to do. These are what these sensors look like. They'll be built in the lab. And these are the current signals, but these are not the signals I would give to a doctor. We take these signals and we run some math and we tell you if it's positive or negative. But and when we take sensor response, these are like the concentrations you would get for each of the 4 molecules that are there. In reality, we only do methyl nicotine, methyl p-anisate because those are unique to Mycobacterium tuberculosis. It turns out the other 2 can come from Mycobacterium avium. So if you happen to have that, it would skew the result. But the point of this slide here is if you look at it, we're getting down to 0.0001 millimolar, detection limit, even more sometimes. The physiological concentration in TB patients that I've measured ranges from 0.1 millimolar to 10 millimolar, depending on when you get what stage they're in. That wasn't in any paper. I had to figure that out. It took me a year to figure that out. No one wanted to publish it. And I can understand why, because the instrumentation is very unique. So even I am not actually publishing it in a paper because, well, these are my instrumentations when it shows, but someone asked me, I say, shoot for this, you'll be around that range. But let's do where the rubber meets the roads. For this specific set of cohorts, these were adults that had TB or were healthy. So I'm going to show you a few slides here. So these were healthy smokers and non-healthy smokers. So the question always comes up, well, what about smokers and your sensor, right? So healthy smokers and healthy non-smokers showed a similar result, at least in 10. Now, some smoke, I don't smoke, but I'm not an expert on cigarettes, but if you look at the ingredients, some of them have nicotine in them. Obviously, some have flavorings like methyl nicotine in them. So it could be possible that you could get a brand of cigarette that might confound this. I want to be clear. There's also things you can eat that could confound this. So you have to think about this very carefully when you take sensor data from patients. Well, that's good, right? So then here you go, like TB versus healthy controls. So here you see a big difference. So at least it's discriminating, at least in this cohort of adults. Now, engineers love to take this piece of data and say it's working. And I point out to them that, while interesting, this is actually not really useful, right? Because the sick person is coming in sick with something. So this is more useful. TB versus non-TB patients. That means they've had cough for 2 weeks, they're congested, and can you tell the difference? So in this particular case, we could, right? With these small cohort of patients these were from Mumbai. And the TB negative and TB positive patients for each of the 2 biomarkers were showing some differences in the signal response. So that's good. Now, you look at this, say, this is great, you've solved the problem. No, I have not solved the problem. My sensors are hard to make. Getting them repeatable and reliable is always a problem. So it's an ongoing process that we're doing this. But this got me my first R01. So yay, it did something positive. So we still work on that. But now let me talk about another version of this that's liquid-based. And this ends up giving you, it's a similar approach, but it's not gas-phase. The other one's gas-phase. This one, I take the gas and I bubble it in solution, and I get an electrochemical fingerprint. So we called it, an electroactive solution-based sensor, and we use off-the-shelf sensors. So this blue screen-printed electrode, I just buy. So I didn't have to make that. All the unique novelties in the solution itself. So it's easier to do, right? Use a similar metal, similar approach, but different format. So the idea here is, if you look at this, is if I have a salt with, in this case, copper in it, and I do something called square wave voltammetry, and I get these peaks, right? And then when I introduce the biomarker of interest, those peaks shift into the present. What's happening is that the metal is binding with the organic molecule inside, so it won't react electrochemically. So it's removing analytes from the reaction. Now, on the left, we have copper, so you see shifts, and I'm going to show you some patient data in a second. On the right, we see cobalt. We actually got a new peak, which means that a new active molecule showed up. So they're both interesting approaches. at first I decided I'm gonna do cobalt, took it to the field and didn't get a good result because it turned out that that new peak was very concentration dependent on how much was coming from the patient, whereas the copper wasn't. So I went back to the drawing board and we just started deploying these. So here's some real data, right? So we get these peaks. So this is a little, I like this approach because I have a little more features to work with, right? So we have this peak 1, we have a shoulder peak, peak 2, and peak 3. So the dotted line here is your blank, it's your reference. You run it, that's what you get. Then you have your patient and you compare it to the patient and you get these new peaks that show up. TB negative patients, they don't differ too much or they don't drop much from the reference. Although we did see this extra shoulder peak show up, but it was at a much higher level. So we could use this data. What we do is we take the distance between the reference to the height peaks in terms of relative current and come up with some conditions, some features for this. So let's zoom in a little bit more here. So peak 2, you can see big drops in the reference peak. I'm sorry, on the TB positive, TB negative. The negative patient is actually above the dotted line, and then the other negative has similar but much different from the TB positive patient. So we empirically found that there are these classifications of signals. What made me happy was that we were able to duplicate these signals in human beings, and so that's always a problem. You do something in the lab and does it work? Is it do something out in the field? Well, a lot of times it doesn't, or it's modified. We actually really get those extra peaks at different concentrations. So I was okay. Then I was more confident about it. So then we took that data, and now we need to use some machine learning approaches. So we do this in the medicine field, right? You take features and you might, it's very famous, right? If I look at different features of, say, a tumor for breast cancer or something, right? Might look at size, morphology, you put it in there, and you use something like random forest machine learning to maybe give you a predictor. So there's papers on that. I don't think it's used clinically, but this is the idea behind using supervised machine learning to do this. So what we did is we have 4 feature sets, and we knew what the patient status was. So we took 75% of the data and trained it and took the other 25% to test it, right? And in this case, for this set of data, we had like 58 patients, 15 TB negative, 40 TB positive, and we ran them through a gradient boosted tree, right? And decision tree. And here's an ROC curve, which to you guys, I don't have to explain. I have to explain this to engineers because they're like, what's this? I'm like, so good part is that at least in this set of data, that you're getting a reasonable performance to a point you're hitting the WHO target profile for sensitivity specificity, at least in the small n. So we have to add more of this, but at least the feasibility is there. And then the nice thing about using this approach is that you can take your features of importance and say, well, which one's most important? And in this case, it actually thinks peak 1 is the most important, followed by peak 2 and shoulder as ties, right? What's nice about this is I can use this to inform the way I do this, and I can take samples and maybe boost the concentration to get a specific kind of peak to show up more. And each one of those peaks really represent an oxidation or reduction state of copper. So it's copper 2, copper 1, copper 0, right? So those are like, and copper 2 is the most common state. It's most stable, but you get copper 1 at lower peak heights, and that's what those different peak heights really mean. It's just available states of copper in solution. So by doing this, though, we get a reasonably good accuracy and a reasonably good performance that shows it has a potential to actually meet the WHO criteria. So if you're to think about this, you flow with healthcare. You walk in, you breathe into a bag, they take the other vitals. This technician runs back. Oops. Let's go back. The technician runs back, bubbles it through, puts it on the sensor and gets a fingerprint, and then they're sick, right? And the idea is that you can do this with something the size of your smartphone and very, very little infrastructure, right? All the other techniques require infrastructure. This doesn't require any of that, right? So the last few minutes, I want to take another tack. So what if you don't want, or you don't have a VOC, but you want to do something out in the field, right? Well, we have this peptide nucleic acid that's electrochemical, right? So what it does, it binds DNA on targets. So you could use microRNA, anything that you want to pull out, and it's reusable. So let's talk about a peptide nucleic acid. So the nice thing about using DNA is that it's really specific. You can get good hybridization. So people love it, right? The downside, though, is that they're not reusable. They're not super stable. So if you're off in Uganda and it's really hot, well, I need to find a good place to store it, right, for it to be stable, right? So there's a lot of problems for this now, but we can use something called a peptide nucleic acid, which is an artificial synthesis, but has a peptide backbone. The reason you would do this is it has a high sensitivity, high selectivity. Most importantly, it's thermally stable. It's not attacked by enzymes either. So that's always a problem. If you ever worked in a biolab before you went to med school, you always have to be careful of having, any kind of digestion of what you're trying to work on, right? But it costs more than DNA now, but only because they don't make them at scale. If you were making them at scale, it'd probably be just as cheaper. So here's a difference, right? So protein backbone, DNA backbone, PNA backbone. And the nice thing about PNA is that it's a neutral charge. It actually binds to things a little harder, a little closer. And you can get very, very selective binding with it. So what we did is we decided that we would take a sequence of Mycobacterium tuberculosis, and my student was clever. He said, well, let me take the whole sequence. Well, everything's mapped now. That's nice. Let me find the sequence that's repeated most often to increase your chances. So he found this 17 base pair length that was repeated at least 16 times. So that way you're not taking your bacteria, lysing it, and just hoping to find something, right? You actually have some intentionality here, right? So 16 times, and then you boost the chance of it binding. And then we did another thing. So the CO there is your complementary DNA. Then a miss is a single mismatch, and then scrambled means it's just not related at all, right? So we throw these all in to see how they change, get an idea about selectivity and signal strength. So these are the electrodes. We purchased them off the spot. So we put the PNA on the surface, and then we incubated it with these different fragments we purchased to see how they perform. So here's some data for. We hybridize it at 50C, and you can modulate the temperature, but it turns out because of the nature of the PNA, you hybrid at 50C. The complementary DNA gave you a current density of 153 microamps per centimeter squared, while the mismatch, which is a single mismatch, was at 80, and then the scrambled was at 79, about the same, right? So it tells us actually that if the sensor is actually pretty sensitive to even single differences, right? That's a big deal. It's a big deal for this. Now, one thing about DNA chips is that you can't reuse them, so you have to keep throwing them out. But because it's PNA, you can heat it up to a temperature where it's stable, but denatures. So we did that. And so we went through these cycles, right? So 95, 25, 30, and I don't know how my student did it. He did it 200 times manually. I feel bad. But you can see that, it drops. But like even between, there's some overlap in the error bars because the general trend that this is actually quite reusable. I mean, if I'm selling these, maybe tell you 10 times and then you sell it, then you can make some money. If I said use it 200 times, I don't know that you're going to make as much, but but the idea here is that you can reuse these. So whatever cost it was to make the PNA, if it was more expensive, you can get back if you can just reuse these and heat them up. So this is a nice novelty of this that allows you to do this. So this is because this has a lot of applications. So I spoke with Shane, and we're thinking about using this for TB of the urinary tract, right? Because the DNA will be there in urine, right? That's the perfect matrix for this. But this can be applied for anything. if you have a microRNA biomarker, traumatic brain injury, it's not a global health issue exactly, but
Oh, it's a huge global
but
health issue,
anything
actually.
you can think of where this would work is useful. So, talk to me if you have some ideas about where to take this. So the potential impact, though, so if we just go back to TB and pneumonia, and then we had others, but ideally what we're trying to do is empower patients and remove these barriers to diagnostics in rural settings. But if you were to look about this as a business person, I mean, the TB global market's going to be $3.9 billion in 2030, $2.8 billion in 2032 for pneumonia, right? So we have to bridge what we make to get out to individual you can use it, right? So this has to be always the case. So there's a lot of potential here. I mean, I'm not going to commercialize them. I'm too busy, but I've been always looking to partner with people to do this, and we have some partners interested, right? But the idea is to really do that. Now, if we think broadly speaking, though, at the U, we really want to create a collaborative ecosystem. That's part of the reason we have these grand rounds. But I sit on the AI machine learning committee for the Price College Engineering. So we call it, I think, the AI hub. We're working different names, but really what we're thinking about is we should have an ecosystem where we use AI and stakeholder engagement principles and take engineering solutions and really try to make a center that does this kind of thing, right? Ideally, we could really actually at the U set ourselves apart from others. There are 7 or 8 major global health centers in the United States, and after Hopkins, UCSF, UC Washington, Minnesota, and they all go to Kampala, Uganda. They have buildings there's literally a building from the University of Washington that looks like it should be on their campus. I can't believe they brought the brick over. It looks like a piece of Seattle's been dropped right into Kampala. And they shouldn't be the only ones, right? They really, and the University of Utah has a we have critical mass of global health here. We just have to coalesce it into something where we can build upon it and kind of bridge engineering with the School of Medicine in all aspects. So let me just acknowledge my collaborators for all this stuff. So a lot of people at the U, that UCSF, McCary University, of course, NIH and NSF and the Gates Foundation have all funded me at some point along the way to get where we want to go. I would be happy to now answer any questions in the last 15 or so minutes, discussion about ideas, collaborations, anything that you guys are interested in knowing. And there's a lot of things in the chat here I should probably.
That was an amazing talk. Bobby, thank you so much for your time and for inspiring all of us. And I'll just say that TB, traumatic brain injury, is and the NIH Fogarty specifically does have R1s for global neurologic disease. So there are some funding opportunities there, and there's
okay, that's good to know. Right.
To think about. And I know that some of our neurosurgery faculty are interested in that kind of stuff, like Carol Budahowski would be interested in that. So he has a funded lab at Utah, but has been in Tanzania doing some work and some training. So there are some potential opportunities. Questions and comments from everybody else. I'm not going to take up all the time here.
Bobby, that's Daniel Young here. That was great. The PNAs, have you looked at whether you can quantify RNA with those?
Yeah, that is actually my next. That is actually literally the next experiment we want to run, is to get complementary sequences for particular RNA biomarkers. So actually, I was going to grab some published sequences for microRNA for TBI, just because I have data as opposed to a random set, and see if that works. That in principle should work, but I I mean, you're better off just making sure that you have the data to show what kind of signals you get. But in theory, that should work. And then, yeah, if you guys have ideas where we want to apply that, I mean,
Yeah, I mean, I think
that one
the,
is wide open.
the quantification and the granularity that goes into quantification is, I mean, even for like nucleic acid based probe, not peptide based probes are difficult, and there's more chemistry and stability issues. But yeah, there's certainly in the area of diarrheal disease, there's certainly gaps in this area. So yeah, good to know.
Because in theory,
think through that.
what happens is if you have these in an array, I mean, the more current you have, the more of those you're seeing. So you should be able to get at least a quantification for concentration for each for the concentration of the nucleic acid you're looking at. So that's been true in other things we've done. It should hold true there, just the way the mechanism in which when they're binding, there's actually, it triggers a current transfer of a molecule that's present that actually adds more current to the system every time that binding event occurs. So it should help.
Thanks.
Jane, you had a question?
Yeah, one's a simple question, and one is more of a just trying to understand how you develop this technology question.
Sure.
The simple question is the VOC versus the TNA, based tests. My understanding is a VOC, you need a live mycobacterium.
Yeah, you need a live organism that, if it's exogenous. Now, if the biomarker is part of an immune response from the host, then I mean it could die and still give you that. So then it would be okay, but you just have to know what you're looking for. Biomarkers from the host in the VOC sense can be robust, but usually there's a lot of them.
Yeah.
So you have to have an understanding of what you're looking for and what you're trying to do. It's hard. I mean, there's a reason that there's no large scale breath tests out there, or even VOC tests. You get it from urine too. In fact, we're about to start a study at Mayo Clinic looking at urine and breath for colorectal cancer, because there's lots of documented literature on this. But while there's some agreement on which biomarkers are common, everyone has a few extra. And maybe that was because that host was different, right? So it's because your cohorts are different. So it's a little bit messy, but you could tolerate it if you can find 3 or 4 that are always there, then you can ignore the rest. It kind of depends on the situation, so yes, that answered the question. Yeah.
And my other sort of more maybe technical question is, in the PNA system, what exactly kind of goes into creating that? Is it similar where it's the reagent that you're creating, or are you creating the PNA chip? Like, I don't quite understand.
Yeah. So we're creating, and this is so I didn't show you other data. So I had created the chip itself, where I have used those same nanotube you saw before. I put them on there and I put gold on top. And the reason I would do that is then I can boost the sensitivity because of high surface area. And I didn't show it here, but I've showed that it's automatically better, right? So that would be super useful if you have low amounts of your biomarker going in. But on the ones off the shelf, I took the gold, and all the novelty or new stuff is in the chemistry to attach it. And then we use a few different electrochemical methods, and some of those electrochemical methods are unique to us, or the conditions or things we come up with. But electrochemistry is that way; it's never an exact science, so it's unique to your situation.
But you're buying the platform with the sort of base.
So yeah. So I buy the platform with the gold on it, and then if I need to make something more high surface area, then I make the whole chip myself, and then I use the same functionalization approach I used on the platform. The nice thing about the screen printed electrodes is that science has been out there for so long that from a deployment perspective, I can buy it and modify it. It just increases your likelihood of success. If you have to make everything from scratch, it's fine, but there's so many more hurdles you have to do to get out there. But I would say, well, why don't you do a 2-phase approach? Take the ones you can buy, get those to work. If they have a performance that is useful for the clinician, great, deploy that, right?
Yeah.
Get the other one ready, and like 2 years later, you have the other new improved version B, and then, whatever, the business people can figure out how they want to monetize that while still making an impact in public health. Any other questions? Well, thank you for having me. I really appreciate the time to hang out and talk to you guys. If you guys want to talk, have ideas for some, I have other tech too. I just showed you 3, but I use hydrogels. I've got a bit of hydrogels. So whatever you guys are interested in learning about, let me know. And then, you know, I have some ideas about how to make some strong collaborations in global health. And I'm going to go see how to get it funded. The crux is we have ideas. The problem is money, right? Right? You're all super busy, right? Like you're so busy, like why would you do this unless there was some money attached to actually support your time, right? And that's not a criticism of anybody. We're all that way, right? We have too many things to do. So hopefully we can figure out some ways to get past that barrier. There's some administration support behind this idea. So if I'm going to shop some ideas around and talk to Sudha and Daniel, and we'll figure some stuff out. But I know we can get there. Okay.
I agree, Bobby. It'd
Awesome.
be great to get people together to think through research questions like this, or come up with new research ideas.
Yeah.
Yeah.
Cool.
Great. Well, thank you
Right.
so much for your time. Thanks, everybody. There are about almost 20 people online. And I'm sorry, I'm out of the country. So
No, thanks for your time. But we had a lot of people in person too. So we're it was a solid showing. It was good.
there's lots of interest and I think we're still forming this community. So I appreciate everybody making time to join. So thank you. And happy holidays. See you next in the new year. So thank you,
Cool.
everyone.
Let's have a good day.
Thank you.
Bye.